That's great. Good afternoon. Welcome to Department of Medicine Grand Rounds. It's my pleasure today to introduce our speaker, Dr. Scott Lippman. Dr. Lippman is Senior Associate Dean and Associate Vice Chancellor for Cancer Research and Care, Distinguished Professor of Medicine, Director of the UCSD Moores Cancer Center, and holds the Chugai Pharmaceutical Chair in Cancer. He's also an adjunct professor at the Salk Institute, the Sanford Burnham Medical Research Institute, and MD Anderson Cancer Center. Dr. Lippman earned his MD degree from the Johns Hopkins University School of Medicine and completed his internship and residency in internal medicine at Harbor UCLA Medical Center. He completed a fellowship in hematology at Stanford University and a fellowship in medical oncology at the University of Arizona Cancer Center. He has participated in the national leadership of clinical and translational research planning and development within the NCI cooperative group setting, and currently sits on the National Institutes of Health Clinical Trials and Translational Research Advisory Committee. He has served on numerous Cancer Center external advisory boards and major trial steering committees, and has played a leadership role in major AACR and American Society of Clinical Oncology committees and programs. Prior to joining UCSD, Dr. Lippman served as Professor of Medicine at the University of Texas MD Anderson Cancer Center. He's the principal investigator on an R01 on targeting signaling vulnerabilities for oral cancer prevention and has authored over 500 original peer-reviewed publications. Dr. Lippman will be speaking to us today about 9p21 copy number loss drives precancer cells to invade, resist immune response, and checkpoint therapy in HPV head and neck cancer. Thank you, Dr. Lippman.
Thank you very much. It's a tremendous pleasure and honor to give this presentation. As was just mentioned, thank you for that very kind introduction. It's wonderful to have the opportunity to present the results of a 5-year multi-institutional project involving 4 NCI comprehensive cancer centers, which identified genetic abnormalities that both drive precancer cells into becoming an invasive type of head and neck cancer, and patients who are least likely to respond to immunotherapy. A key and unanswered question in tumor biology is how a cancer escapes recognition by the immune system, evolving from immune hot to immune cold tumors, the latter often impenetrable by immune cells. The results of our studies indicate that for HPV negative head and neck cancers, the answer lies on the short P arm of chromosome 9. Here are my disclosures.
Dr. Lippman, can you move your head up a little bit so we can see your face more?
Oh, sure. Is that better?
Beautiful.
Sorry. So here are my disclosures. We began studying the invasive disease transition in this particular disease, where in this cartoon, you can see precancerous cells evolve from hyperplasia to increasing dysplasia and carcinoma in situ. But at some point, these abnormal neoplastic cells invade the basement membrane and develop the first of 2 lethal transitions in the development of epithelial cancer. And we were interested in studying this process. There had been a model put out a few years ago, maybe about 5 years ago, that by Angelica Mann and others that suggested that the immune system, the host immune response, could actually suppress, as shown by these inhibition arrows, the pre-invasive stages of epithelial tumorigenesis. But at some point, when these cells invade the basement membrane, they then flip the switch, and now the neoplastic cells can inhibit the host immune system. So we wanted to study this process in HPV negative head and neck cancer. The data that existed at the time we began in 2017, 2018, 2019, was that experimental cell systems trying to create a precancer system artificially by techniques such as microcell transfer and other techniques to artificially induce karyotype abnormalities, copy number, chromosomal copy number abnormalities, or aneuploidy. And when this was done in normal cells, often using retinal pigment epithelial cells, that this was recognized by the immune system and triggered an immune response. On the other side, there was data coming out, again, about the same time. This is from Teresa Davoli's group, published in Science in 2017, where in human cancers, not experimental systems, but in human cancers, several cancers, notably head and neck cancer, had the opposite phenomenon. Those that had high levels of chromosome copy number alterations or aneuploidy actually had immune cold tumors, his cytotoxic immune score, with L is for low and H is high copy number alterations. So it appeared that the same sort of copy number alterations that triggered an immune response in precancer may actually trigger immune evasion, immune cold tumors in cancer. And so we wanted to study this in people. The other interesting bit of data that was emerging about the same time was the fact that, at least in a number of squamous tumors, showing lung squamous cancer and carcinoma in situ, lung squamous cancer is very similar computationally in terms of copy number patterns to HPV negative head and neck cancer. But what people have found, for instance, in the circus plot, the outer rim being the lung squamous cancer and the rim right next to it, the in situ disease, where red is copy number gain and blue is loss, you can see here that they tend to track together. For instance, chromosome 3p, which is often involved in this disease, you can see 3p is lost, it's blue in both the cancer and the precancer, and the 3q is gained. This is a plot from Ledmil Alexandrov here in this same setting, where losses are in purple and blue and gains in red and orange. The blue is cancer, the purple here is the precancer, and the 22 chromosomes, autosomes, are at the bottom here, showing the arms. And basically what you see is that the purple and the blue really are closely aligned. There's not a big difference between copy number alterations in the squamous precancer compared to the cancer. And that led to the idea, together with other data, that maybe it's not an additional genomic event, copy number event, that drives the transition from precancer to invasive disease, but perhaps it's an immune event. And this is just some data from Sam Janes. There's a couple other reports that suggest this is a lung precancer, and this is progression of the lesion and regression of these precancer lesions, cytotoxic T cells on the vertical axis. And these are the kind of patterns that seem to exist, that in lesions that progress to invasive cancer, they tend to be immune cold, associated with a low tumors cytotoxic T cell count, and tumors that respond, cancers that respond, tend to be immune hot. So these were the data going into our study of oral or HPV negative precancer. And we studied all of the sites of reported recurrent copy number alterations in this disease. The first report we published about 25 years ago now, looked at 2 recurrent copy number loss in oral cancer and HPV negative head and neck cancer, loss of heterozygosity at 3p14 and 9p21, and where you have oral cancer-free survival here, and time in months on the x-axis. And you can see that loss of one or both of these particular chromosomal sites were associated with a worse outcome, a lower cancer-free survival than those that had retention. This has been confirmed by a number of groups, and we also confirmed this prospectively, where in the same Kaplan-Meier type plot, the black are patients that have loss of heterozygosity at any of these multiple sites had a worse outcome than those that had retention. And a lot of work, including Trey Eidecker here, in collaborative work, we found that chromosome 3p seemed to be, if anything, the most important recurrent site of loss in this disease. But this turned out not to be the case in the case of the immune events. So we studied all of the reported sites of recurrent copy number loss on 7 chromosomes in this particular study. The design is shown here. So we looked at a prospective oral precancer cohort and a cancer cohort, and we tried to look at the same sorts of copy numbers and immune metrics. In precancer, these lesions are very small, so it's hard to do whole exome sequencing, although I'll show you some data towards the end. So we used PCR here and looked at 3p14, 9p21, 70p13, and the others shown here. And we looked at these immune cells, particularly CD8 cytotoxic T cells. In the cancer setting, we tried to parallel this despite the different methodology of whole exome sequencing. We looked at the same 3 arms, predominantly in immune markers, and incorporated a number of cell lines. And this is what we found, and shows sort of the design. So on the left side of this slide shows the anatomic location of the most prominent site of HPV negative head and neck cancer, and that is the oral cavity. The upper middle H&E images show the progression of cancer to carcinoma in situ, followed by the first of 2 pivotal events in the development of lethal epithelial tumors. The orange arrow and question mark show the focus of our initial interrogation, that is to study the previously unknown biology behind the invasive disease transition. And you can see here these very abnormal neoplastic cells here in carcinoma in situ, but you see the basement membranes intact. But then something other than a key genomic event seems to cause obliteration of this basement membrane, and you can see here the cells, neoplastic cells, just flood through the basement membrane. And the HPV negative head and neck cancer and its precursors are characterized by a number of well-studied recurrent sites that I mentioned below and referred to this figure. So this is what we found initially. There had been considerable debate on the various sites of copy number loss in this disease, and whether, in fact, the specific regions that were lost were important, or whether it was just the loss of heterozygosity event that was a surrogate for chromosome instability. And we and others went in with the former, or more specifically, that we felt that loss at any one of these sites might be the key event, and there wasn't a specific region that was driving this, and part of it's because of the focus on chromosome 3p, which the 3p14 region doesn't have any identified tumor suppressor genes. But what we found, surprisingly, is that one site on one arm was very different than all the others, and that is chromosome 9p21.3 loss produced a very different effect, and showing this in the sort of cancer on the left, precancer on the right. We first saw that 9p21 loss triggered an immune response, shown here as immune hot. This is with CD3 cells, but I'll show you CD8 as well. So in the precancer setting, this specific copy number alteration did trigger an immune hot, which is consistent with the preclinical work I just mentioned. In cancer, it was very different. And so I'll show you data that loss of heterozygosity at this region induces a immune cool signal, if you will, which becomes profound in the setting of advanced disease and with concurrent p53 mutation. Talk about deep deletions and the effect of deletion size on the immune microenvironment. So this is what we found in cancer. We found that loss of this focal region produced an immune cold signal. Wasn't quite significant, 0.06, but it was very different than every other site that we looked at. And again, completely unexpected. We then looked at this by stage and found that in high-grade precursors, the 9p21 loss is in this blue color and wild type is in red. You see really no change in loss. If anything, there's an increase in CD8 T cells in the precancers that lost 9p21. When you cross over and look by stage, you see that there does seem to be progression, particularly as you look from stage 1, early stage, to stage 4, 9p21 loss of heterozygosity produces a minimal effect in stage 1. Again, this is 9p21 wild type versus loss of heterozygosity, and CD8 counts on the y-axis. No big difference in tumors, early stage tumors that were wild type or had lost this region. However, when you get into more advanced disease, this is stage 4, you see quite a strong effect of loss of heterozygosity at 9p21, producing strong immune cold tumors. So immune cool overall, but in advanced disease, it was much stronger. And then the other finding was that the size of the deletion around 9p21 seemed to be very important. Whereas focal loss of 9p21.3 seemed to be necessary, larger deletions encompassing other regions, 9p21 and other regions on the arm of 9p drove profoundly immune cold tumors. This is shown with CD8 T cell levels. And then we also looked at various signaling pathways. In cell lines, there was a 9p21.3 loss effect on suppressing cell intrinsic SASP, and we saw effects on some of the immune pathways that I'll talk about, and I mentioned p53. So at this point, we went from really focusing on the invasive transition in precancer to heavily focusing on cancer in a number of different cohorts and cell lines computationally. And so this is what we found. We found that in addition to the surprising finding that 9p21 seemed to be very different than all the other sites of copy number loss, when we looked at all of the chromosome arms, losses and gains in HPV negative head and neck cancer, only the loss of the entire chromosome 9 or 9p produced an immune cold signal. It's a cytotoxic immune score, and if the orange bar is above this line, statistically significant. So in head and neck cancer, at least in HPV negative disease, 9p seemed to be a much more important driver of immune cold tumors. We think that the chromosome 9 loss effect was largely driven by 9p because we didn't see an effect with 9q. And so we think that this is what's happening. If you imagine going from left to right with precancer on the left and cancer on the right evolving, 9p21 loss seems to be a really important early event. A lot of interest in CDKN2A located at this site. But as you progress to early stage and advanced stage cancer, what we think is happening is that the deletion size, shown in dark blue, increases substantially around this region. So instead of just focal loss of 9p21, it appears that there's loss of regions on both sides, larger deletions encompassing 9p21. And this was interesting to us because when you look at what's on chromosome 9p, so here's 9p and 9q, it turns out that chromosome 9p is a relative hotbed of immune regulatory genes. So on 9p21, again, you not only have CDK 2A and 2B, but the entire interferon alpha gene cassette is located at 9p21. It's about 16 genes. And also the other region that's getting a lot of attention is 9p24.1, which is the location of JAK2, which is a very potent interferon gamma pathway gene. Tony Rebus and others have shown that alterations, mutations, and loss of JAK2 can drive resistance to anti-CTLA-4 drugs in melanoma. But also located at 9p24.1, you have both ligands of PD-1. So you have CD274, which is the gene that encodes PDL1, and you have the gene that encodes PDL2. In addition, through a lot of studies looking at cell lines and cancers and different filters, we identified a region that hadn't been noted before at 9p13 that included a 21 gene cluster of genes that seemed to potentially play a role in antigen processing and presentation. So we're continuing to look at this arm very deeply, but it appears that for some reason, the vast majority of immune regulated genes in human chromosomes is located on the short arm of chromosome 9, which raises some interesting evolutionary aspects to it. But this seemed to be one of the aspects that came out of this and related work. And then I mentioned the p53 association, loss of heterozygosity at 9p21.3 in the context of p53 mutation drives immune cold tumors. And here we're looking at stage 1 and 2 patients, CD8 T cells on the vertical axis, and the p53 mutant status on the x-axis. So this is wild type and this is mutant p53. And again, the 9p21 LOH is in purple and wild type in red. And in early stage wild type tumors, you don't see a significant difference of or effect of 9p21 loss on immune hot or cold tumors, but in p53 mutant tumors, 9p21 loss is a very strong driver of low CD8 infiltrates immune cold tumors. The reason we looked at stage 1 and 2 is that in advanced stage of HPV negative head and neck cancer, the p53 mutation rate exceeds 80%. So it becomes very difficult to see an interaction here, but there's some experimental data that would suggest that 9p21 loss is necessary, but may require mutant p53 to drive immune cold phenotypes. So this is what we at this point did, focusing on head and neck cancer. And just to show you here, this is HPV negative and positive disease. For some reason, we found that the 9p21 loss seemed to be focused mostly on HPV negative disease, very low rates in HPV positive head and neck cancer. When you look at several of the key sites, this is just one of the main figures looking at some of the major sites of loss, copy number loss in this disease. In this case, looking at pathology correction with CD8 T cell infiltrates, you see that by far the strongest driver of immune cold, low CD8 infiltrate tumors is 9p21.3 loss, regardless of the deletion size. We didn't see that with 3p14 or the other sites. Overall copy number alteration or aneuploidy had been reported by Devoli's group, who we collaborated with on this study, as being a very strong driver of immune cold tumors. I showed you that figure early on, especially in head and neck cancer. And in this study, we confirmed that overall aneuploidy was in fact an important driver of immune cold tumors, but the 9p21 loss was more significant and independent of this non-specific marker. And when we looked at many different cutoffs and computational approaches and analyses, this association between 9p21.3 loss became stronger and stronger. This just shows with purity correction using absolute. The p-value is even stronger, continuous, using 5 or 10-fold cross-validation. In every analysis that we did, the one consistent finding was that 9p21.3 was driving immune cold tumors. We also found that that 9p21 loss was associated with suppression of immune activation markers such as interferon gamma and granzyme. I mentioned the pathway suppression. Another really surprising finding was that 9p21 loss tumors had profound suppression of CXCL910. This is a chemokine family that actively recruits T cells to tumors. We looked at 17,000 molecules that are altered with copy number changes in this disease, and found that CXCL910 stood out by far from all the other associations. Again, this was very exploratory. But with FDRs in the 10 to negative 15 or 10 to negative 20, this was a very striking effect compared to the others. But again, it was not hypothesis based, and so we didn't know what to make of it. And then within the same period of time, a couple months earlier, Charles Swanton came out with a paper looking at predictive markers of benefit in 7 solid tumors. One was head and neck cancer, and looking at PD-L1 and TMB and CD8, the usual suspects. But in head and neck cancer, the strongest predictor of benefit for anti-PD-1 checkpoint therapy was CXCL9. So this was really intriguing because we found that 9p21, which produced profoundly immune cold tumors, the greatest effect was on suppressing CXCL910. But again, it was exploratory. But then a couple months after this work had come out, another study from MD Anderson came out a couple months later, and it was a pan-cancer study, but reported very similar effects in head and neck cancer. So they also found that the 9p21 loss phenomena was primarily an HPV negative phenomena, not HPV positive disease. And they looked at a number of different tumors. This is HPV negative head and neck cancer with CD8 levels on the y-axis. And this is 9p21 wild type. This is loss in one allele, and this is a so-called homozygous deletion. And looked at it in lung adeno and pancreas and many others, and found very similar results that we reported in head and neck, that comparing 9p21 wild type to loss, there was a very strong suppression of CD8 T cells with the Q value shown here. There was not a big difference between so-called heterozygous and homozygous deletion, which I'll come back to, but in any case, there was a clear effect, which was seen in several other tumors. And provocatively, they also found the same thing regarding CXCL9 and 10, that 9p21 loss tumors had profound suppression of this. So within the span of 9 months, there were 3 studies that found very consistent findings involving 9p21 and CXCL910 to suggest that CXCL19 suppression may be a very important mechanism for how 9p21 suppresses CD8 infiltrates. CXCL9 and 10 are not located on chromosome 9. We brought in Steve Dubinet from UCLA, who's the world's expert in chemokines, to look at this, but we still don't know what the mechanism of how 9p21 suppresses these chemokines. But one thing that's interesting is that you can bypass this by directly injecting these chemokines. And Steve Dubinet had a spin-off company that's actually injecting chemokines into tumors that have low levels to see if they can bypass whatever the mechanism is of 9p21 suppression of these molecules. So this set up this hypothesis, the data that I showed you. This is showing overall high levels of aneuploidy or copy number alteration, and this is showing at 9p with the classic immune hot and cold phenotypes with if you look at CD8 cytotoxic T cells with immune hot, you can see the T cells are infiltrating the tumor. In immune cold tumors, for various reasons, these T cells do not seem to recognize the tumor and can form a rim around the tumor and escape this disease. So there's really two types of solid tumors in the immunotherapy world. That's immune hot and cold, and generally immune cold tumors are resistant to immune checkpoint therapy or other types of immunotherapy. So when we saw that 9p21 suppressed T cell infiltrate, produced very strong immune cold tumors, then we hypothesized that if this genomic alteration can cause immune cold tumors, would it identify patients that might be resistant, less likely to respond to immune oncology approaches such as immune checkpoint therapy. The interesting thing in head and neck cancer about this is that 9p loss is likely to have the greatest impact in patients who have recurrent metastatic head and neck cancer, because 80% of those patients tend to be HPV negative. And as I mentioned, 90% of the deletions occur in HPV negative patients. So, so we went on to look at that, and just to orient people who don't do a lot of head and neck cancer. Head and neck cancer is the sixth most common cause of cancer worldwide. HPV positive head and neck cancer is increasing substantially in the United States and other developed countries. However, HPV negative head and neck cancer still is the most common and by far the most lethal type of head and neck cancer in the United States and globally. What's interesting about this is that despite the striking differences, again, comparing HPV positive and negative head and neck cancer, the striking differences in etiology, biology, genetics, and more recently, even the staging system, the AGC staging system has changed at the last iteration for HPV positive disease. The outcomes with immune checkpoint therapy are very similar in HPV positive and negative disease and tend to look like this. This is actually the Keynote 40, one of the registration trials for immune checkpoint therapy in head and neck cancer. Ezra Cohen was the PI, lead author on this trial, and second line therapy. This is Kaplan-Meier with survival percents on the y-axis and time on the x-axis. And pembrolizumab, which is an anti-PD-1 drug, is shown in red, and then standard of care, generally chemotherapy, is shown in blue. And you see that clearly the pembrolizumab patients had improved survival compared to standard care. It's not dramatic, but this is really a first in this disease and shows that it is potentially possible to cure these patients. About, as I mentioned, 15% will have durable response in treatment of unselected patients. This particular survival curve looks very similar to many other solid tumors where there are FDA-approved indications for immune checkpoint therapy. Different types of lung cancer, liver cancer, cervical, bladder, esophagus, gastric, many cancers where there are FDA-approved anti-PD-1 or immune checkpoint therapy have a survival curve that looks like this. About 15% of all comers, give or take, will have durable responses and presumed cure. The clear exceptions to this are in melanoma and renal cell cancer, which have higher rates of response and durability. It's extremely important to be able to identify patients that might have primary resistance, this 85%. And particularly, for instance, in second line head and neck cancer, where median survivals or mean survivals are about 6 months. So a course of, and there is no companion diagnostic for this. PD-L1 is not a companion diagnostic for head and neck cancer. So pretty much we treat all patients in recurrent disease. 85% will become resistant, will have primary resistance at huge financial and quality of life costs, severe toxicities, and most importantly, losing valuable time to try other therapies. We've shown now in others that patients with 9p21 loss actually have a better survival with chemotherapy compared to immunotherapy. So it's extremely important to be able to identify these patients to enhance the chance of identifying people more likely to respond, less likely to be resistant, so that they can have other therapies. Again, PD-L1 is not a companion diagnostic, although it is used. Most PD-L1 positive patients do not respond, and tumor mutational burden, at least with the cutoff of 10 that the FDA used, doesn't seem to be as predictive for head and neck cancer. So it's on this background that we hypothesize that, as with targeted therapy, tyrosine kinase inhibitors, for example, could genetic information or genetic lesions provide predictive information for immunotherapy. This is a busy slide, but just to kind of show you what's emerged. So initially, we studied this question in a cohort of patients that have been treated with anti-PD-1 drugs, either Nivo or pembrolizumab. And it's a little hard to see, but on the left is our patients who did not receive immune checkpoint therapy, and the right patients received anti-PD-1 drugs. And what I hope you can see, the blue is wild type and the red is loss at 9p, is that in the non-immunotherapy treated patients, there was no difference in survival in patients who had lost 9p21 or who had retained. The hazard ratios and p-values were close to 1. But in patients who had received anti-PD-1 and checkpoint therapy, the curves differed. In blue are the patients with no loss, and they had durable response rates of about 30%. And those that had 9p loss did poorly. So these data suggested that 9p21 loss, 9p arm loss, may in fact be a specific predictive marker for precision therapy, at least in HPV negative head and neck cancer. And the other study I mentioned from MD Anderson, it came out afterwards, looked in many cancers and found provocative results. This is lung adenocarcinoma, where PD-L1 IHC is a companion diagnostic. And so again, typical Kaplan-Meier plot with percent alive on the x-axis and time on the bottom. And what they're trying to show is the dark green, is our patients with high PD-L1. And we can get into how that's defined. That's one of the challenges with PD-L1, of course, is there are different ways to measure it and antibodies and metrics. But in any case, those that had high PD-L1 had a better survival than patients with low PD-L1 in the light green. But what they were trying to make the point of is, if you've lost 9p21, which is in red, it didn't matter what your PD-L1 was, all the patients died. And again, we'll come back to this, but one of the challenges with PD-L1 in this setting is that chromosome 9p24 encodes the gene that makes PD-L1. So the question is, how much of the effect that we see in 9p loss is due simply to a decrease in CD274 and therefore PD-L1. These are two other studies that came out subsequently earlier this year, and similar types of patterns. These are mixed patients of different types. But in any case, the response rates seem to be higher in patients who were wild type for 9p21 compared to those that had 9p21 loss. This is another study from EBOT looking at a number of different cancers, different surrogates. This happens to be non-small cell lung cancer. And again, a worse outcome for patients who had surrogate loss of genes at 9p21. The interesting thing that emerged with this kind of obsession with 9p21, which seemed to be there from the beginning, was the fact that is 9p21 the answer, the only answer. And there was data like this that came out. This was actually with the MD Anderson paper, the Han et al., where they looked at PD-L1 IHC expression based on whether the tumor had lost 9p21. And what they found is that in tumors that had lost 9p21, the expression of PD-L1 protein was very low, much lower than in wild type. So this led this group to speculate that it was highly likely that there was co-deletion of 9p24.1, that patients with loss of 9p21.3 likely had deletions extending to 9p24.1, and that would account for this difference in PD-L1 levels. If it was just 9p21 loss, you wouldn't expect to see this. And so they speculated on this a bit in their discussion, but they didn't actually look at the data this way. So we decided to look into it in a little more detail. And in fact, as I'll show you, showed that 9p24 does seem to play an important role in the predictive value of anti-PD-1 drugs in head and neck cancer. And I'll show you that. This is hard to see. It's looking at depth of deletion. This is another thing that emerged. So the things that sort of came out is that 9p21 focal loss produces immune cold signal. It's stronger in advanced disease. It's stronger in p53 mutant tumors. And that deletion size, particularly encompassing 9p24.1, seemed to be important. But there also was a school of thought emerging that the depth of deletion was important. And that is that loss of 9p21 on one arm of a diploid tumor versus on both arms. And, so that's where you get to using terms like homozygous deletion. But as everyone here knows, that tumors are not diploid generally. In fact, we found that HPV negative head and neck cancer was closer to triploid, 2.6 or 7 copies. So homozygous deletion is hard to know what that means. But in any case, we looked at this using the same methods from this prior paper, and a separate method calculating depth of deletion, controlling for ploidy. And basically what you see is, and I showed earlier, is there's just not a big difference. For whatever reason, loss of 9p21 on one arm seems to be enough to cause the effect that we see on suppression of CD8 T cell infiltrate. That further loss, of more copies doesn't seem to play a bigger role, at least not in head and neck cancer. It's hard to know why. There's a phenomenon called haploinsufficiency that Webb Cavaney has really discovered in his work with LOH. But anyway, in reality, there was not a big difference when you had deep deletions, again, at least in head and neck cancer. And then I'll show you some new data in precancer. So based on this, this finding here that suggesting that larger deletions may be important, we looked at 9p24. This is a collaborative study led by Ezra Cohen and Sylvie Gutkind and Ludmell, a number of people here, as well as groups at NYU. Again, trying to dissect this 9p. And although there's interest in other regions like 9p13, the two regions that really have emerged over the past year as being important are 9p24 and 9p21. Each of those have 3 bands. So the key ones are 9p24.1 and 9p21.3. And you can see some of the genes there. And in this study, we did whole exome as well as whole genome sequencing. I mean, as whole transcriptome, excuse me. And what we found is interesting, a couple things. We found that, again, at least in head and neck cancer and maybe other squamous tumors, when you look at the frequency, this is chromosome 9p, where you have gain on the top and loss in purple frequency. And then you look at CD8 T cells or immune score. What was interesting is that the same regions of 9p loss that produced immune cold tumors also showed a very strong gain immune hot signal, which is a little unusual that the same side would be both. And this seems to be more limited. The 9p loss immune cold signal seems to involve a number of different tumor types. The gain part may be limited to squamous tumors. But you don't see it in adenocarcinomas, for instance. And the other thing to note is that in this new study that should be coming out soon, hopefully, we did look at other tumors, not in the same sort of depth, but we found, for instance, that pancreatic cancer had a very different pattern. You can see that it's pancreatic cancer with 9p loss, and there is absolutely no 9p gain. No 9p gain hot signal in pancreatic cancer. So, very different patterns. And, that's why we focused really on head and neck for reasons I mentioned. The other thing we found is that not only is the gain interesting and different than everything else, which had been 9p21 loss, immune cold, resistant story, that the gain may predict benefit. And this is actually something that Jack Bowie really pointed out. We were talking about some of the CAP-CLIA validation of this, and he noticed on this graph, we were focused entirely on what everyone else was, that 9p loss was cold and resistant. And he said, well, what about this gain? And it was staring us right there. And after that, we went back and he was right that, in fact, gain predicts immune hot, again, in head and neck, and looks like it predicts better outcomes. So if you look at 9p24 now, 0.1, at high levels of expression, 60% or above, the blue are the ones that got immune checkpoint therapy and had 60% or greater expression. This is their survival now. So it's not just a resistance in terms of predicting survival. In contrast, patients treated with immunotherapy that had 9p24.1 expression less than 40 or 60%, they actually did worse than chemotherapy, which is shown in the middle here. So, I think it's getting to the point where, we're able to, at least through sort of loss and resistance and some of these gain patterns, hopefully get to where we can better select patients that are more likely to benefit from this. This is just a reminder to mention that after the initial two reports, the one from our group and the one from Han group, after these two reports, the 9p loss became CAP-CLIA certified and is now a CMS reimbursable test that we can order on our patients to better help select therapy. This is just the resistance part, and 9p loss occurs in about 20% of recurrent head and neck cancer patients. The expression data are being CAP-CLIA certified now. And then finally, I'll show some very new data in precancer, sort of coming full circle. These data were presented at ESMO, the big European meetings, global meeting in August by Glenn Hanna, but the genomics were not presented. I'll show those here. And basically, this is a study led by Glenn Hanna and his colleagues at Dana-Farber, studying a very high risk oral precancer, HPV negative precancer. This shows the lesion in different parts. The risk of developing cancer in this group is about 50% at 5 years. I mean, it's unheard of. And so this was the design shown here. This is a clinical protocol with nivolumab was the active treatment. And the primary endpoints were response and cancer development. And these are the results. So In general, it is a single arm study. This is the first study I'm aware of, actually, of any immune checkpoint therapy in any precancer setting. So really remarkable study that Glenn's group led. But these are just pictures of pre and post, and extremely well documented response rates. The outcome in the single arm study of about 30 patients was far better than what one would expect. For instance, the development cancer was about 20% instead of 40%. It looked very promising, but it was a single arm study. So in this waterfall plot, you can see the patients here that had progressed, and then you see patients who responded clinically but developed cancer. And here you can see how long patients were on treatment, and with the asterisks when the cancer developed. This is the overall cancer-free survival curve, which is far better than one would anticipate in this disease, but again, needs to be shown. In this study, PD-L1 had no effect on outcome. The hazard ratio was 1. And the genomic data are here. And this was an ad hoc study that Glenn had reached out to us to look at based on some of the studies that I mentioned earlier in HPV negative head and neck cancer and precancer. And this is the bottom line. There were 6 patients who developed oral cancer, and then there were 16, I guess, or 14 that didn't. Every patient who developed invasive cancer had 9p21.3 loss, every single patient, compared to a lower rate in those that didn't have cancer. And, there's interesting findings within this. For instance, case 1 had 9p deletion. I was going to show the plot. I couldn't find it. But in this case, this patient had a large deletion, including 9p21 and 9p24, and developed cancer in a short amount of time, had progressed clinically, pathologically, and developed cancer in less than 2 months, and had a large deletion that included 9p21. So this is really kind of exciting. The big pharma who sponsored this is now putting together a large randomized trial to extend these results, and 9p21 will be the only upfront primary stratification factor. It'll be led by Dana-Farber and UCSD, as well as several other centers, and that's being developed. So I will stop there. I just want to thank everyone. This has been incredibly exciting study, on a personal level, having been in this field for a long time. It's exciting to be part of studies that actually may be able to help people. In this particular case, the finding was a complete accident. I wish we could have said that we were looking for this 9p, but because of the nature of the group, Ludmill, Alexandrov's lab, Hannah Carter, other major groups, Ezra and so on, we were able to recognize this and build on it. And it's just the beginning. And maybe this other study from Anderson is right, that this finding will be helpful in other types of tumors as well. But again, it's certainly exciting when you're in clinic and patients are wanting to know, what are my chances of responding to immune checkpoint therapy, which of course is transformative, but there aren't great predictive markers. So, and I also want to credit the Precancer Genome Atlas that this is part of, and there are a number of people at UCSD that are playing key roles in that. But again, here are the key people from UCSD, Ludmill's lab with 2 postdocs, Teresa DeVoli's group at NYU, Glenn Hanna, Steve Dubinet, people at Anderson and others. So I will stop there and I'd be happy to take any questions. Thank you very much for your attention.
Thank you so much, Dr. Lippman, for that very thought-provoking presentation. We have a question from one of our chief residents, Dr. Tong. Fantastic talk regarding the tumor infiltrating T cells in 9p loss tumors. Is it known whether those CD8 T cells that are able to infiltrate are dysfunctional or exhibit an immune exhaustion phenotype? Also curious what TILs appear to be excluded at the periphery of tumors, so also wonder if other microenvironment components such as tumor stroma, regulatory T cells, or myeloid cells may be helping in TIL exclusion.
Yeah, so those are really very insightful questions, and the easy answer is we don't know the answer to those. Where all of this is really pretty new, you know, it's been a roller coaster over the past year sorting through this, but we're very interested in other immune effects, and particularly on macrophage biology, and have been in touch with Judy Varner, who's, you know, a or the global leader in macrophage immune oncology, and to set up some experiments to look at that question. She's very involved with Ezra and Sylvia and Joe Califano as part of the Head and Neck Explorer. So, you know, I think CD8 cytotoxic T cells tends to be the prototype for immune hot cold and infiltrate, but and we didn't see these patterns when we looked at CD68, which of course is a non-specific marker for macrophages. But I think, you know, these kind of questions to dig deeper is what we're all really excited about doing, and really, you know, plays on the tremendous strength here in Andy Goldroth and really top, you know, immunologists. So Steve Schoenberg and everyone. So we're hoping to really, you know, go into the next level and see what other cells, look at T cell exhaustion, look at different macrophages and so on.
Thank you, Dr. Lippman. From Dr. Tanaka, when there is such clear evidence of the role of a chromosomal deletion like 9p loss, how much of a role does epigenetics play?
So, yeah, again, we don't know. We haven't looked at that. You know, the data that we saw with p53 mutation, for instance, has raised the question of, you know, there are patterns that are intriguing with p53 wild type, and it could be that there's no p53 mutation, but there are downstream signaling alterations to p53. I don't know if there are epigenetic causes of 9p21 loss. I do know that Adam Engler and Melissa Jimrick and others are interested in germline alterations in 9p21.3 different SNPs to see if they, you know, if there would be some sort of interaction between germline 21 SNPs and the somatic changes. But you know, we've met on those, but we haven't really started those. And again, Hannah Carter's looking at the germline aspects of a lot of these immune effects. But I'm not aware of any epigenetic associations or effects on 9p21 or 9p24 expression.
Thank you so much, Dr. Lippman. If there are no further questions, I want to thank you again for this outstanding presentation. And I hope that all of you will join us again next Wednesday at noon for another Department of Medicine Grand Rounds. Have a nice day. Thank you.
Thanks, everyone. Bye. Good day.